In this video, we will learn about the last characteristic feature of fungi, that is, different methods of reproduction. Ms. Alina Agnihotri of Puran Chandra Vidya Niketan, Kanpur will tell us more about this topic.
We come to the last characteristic feature, that is reproduction in fungi. Fungi are those group of organisms in which all the three kinds of reproduction take place, that is vegetative, asexual and sexual. I hope you are clear with what is vegetative, what is asexual and sexual kind of reproductions. But for a recap, in vegetative reproduction, there is no sexual gamete involved. It is done by breaking up or by division of the cell body or the hypha or mycelium of the organism. Asexual reproduction is done by certain small spore-like structures which are of various kinds, and sexual reproduction involves the fusion of two different kind of gametes. But since fungi is in the lower group of organisms, we do not find male and female filaments as such. There are certain filaments which behave as a positive stained filament and a negative stained filament. As a result of the fusion of these two, sexual reproduction is carried out. We come to vegetative reproduction. Vegetative reproduction is done by three methods. The first method is fragmentation, second budding, and the third is fission. Fragmentation means that a filament can break from any part and form two different kind of organisms. That is, example is rhizopus. Then in budding, which takes place in yeast, we find that the normal mature cell of the yeast, at some stages when the cell is fully grown and matured, it gives out a bud in which a part of the nucleus is moving along with some cytoplasm. This bud grows in it and accumulates cytoplasm, and when it is mature, it can behave in two ways. Either it remains attached to the filament or to the cell, mother cell, or it can separate and become an individual yeast cell. If it is attached to the mother cell, it can form a chain of cells, and if it is individually, then it can form an individual organism. Then we come to the third method, that is fission. In fission, the cell elongates and the nucleus also divides into two along with the cytoplasm, and two separate cells are formed. These two processes, the budding and fission, they take place in yeast. Then we come to asexual reproduction. Asexual reproduction is done by various kind of spores. They may be endogenous spores, that is, they are formed within any kind of a fruiting structure or sac, or they may be exogenous spores found on a fruiting body outside or attached externally. They are of various kinds: conidia, sporangiophores, zoospores, and it depends on the kind of the fungi in which they are present. Then we come to sexual reproduction. Sexual reproduction is taking place by the fusion of different kind of stains which are present on the filament. The filament at the time of sexual reproduction behaves as a positive or a negatively stained filament, and all the different kind of filaments, they undergo these three stages in the process of sexual reproduction. First is plasmogamy, second karyogamy, and the third meiosis. Plasmogamy is said to be the fusion of protoplasm. When two cells which are of different stains, they come close together, either a conjugation tube may be formed or cell to cell attachment will be there. The cytoplasm of the male cell, that is the positive stain cell, will move into the female cell along with the nucleus. This process, that is movement of protoplasm, is called as plasmogamy. After plasmogamy, either the nucleus are going to combine together, fuse together, and form a diploid condition, that is 2n stage. During plasmogamy, the filaments are n plus n, that is haploid. When they fuse together in karyogamy, they will form a diploid stage, that is 2n, or the nucleus may remain as such next to one another and forming a structure which is called as a dikaryon. And the phase is called as a dikaryotic phase. So a dikaryotic phase is different from a diploid phase. In a diploid phase, the nucleus, they fuse together to form a diploid nucleus, whereas in a dikaryon phase or a dikaryotic phase, the n plus n, the nucleus, they stay together, close together, but they do not fuse. The fusion takes place just before the process of meiosis is to be carried out. After the fusion process, that is karyogamy, the third stage is meiosis, where the diploid 2n nucleus, it divides into haploid spores, and this is found in ascomycetes or basidiomycetes. The spore which is formed is called as either the ascospores if they are formed in ascomycetes, or basidiospores if they are found in basidiomycetes. Later on, these asexual spores and the sexual spores, they germinate on suitable substratum under suitable or favorable conditions and give rise to a new mycelium.
Vegetative reproduction can take place by fragmentation, fission and budding. Asexual reproduction is by spores called conidia or sporangiospores or zoospores. Sexual reproduction is by oospores, ascospores and basidiospores. All the different spores are produced in a distinct structure called as fruiting bodies. Plasmogamy can be defined as the fusion of protoplasms between two motile or non-motile gametes. Karyogamy is nothing but the fusion of two nuclei, means production of diploid cell. When diploid nucleus is divided into haploid spores, it is known as meiosis. These asexual and sexual spores will grow on the suitable substratum to form a new mycelium.
For more such videos, log on to CBSE section of jagranjosh.com.